There is no light here, and there has never been light at this depth, not since the ocean formed 4 billion years ago from water vapor condensing out of Earth's primordial atmosphere. Sunlight, that fundamental driver of almost all life on Earth's surface, penetrates only about 200 meters into even the clearest ocean water, before being completely absorbed, scattered, transformed into heat by the water molecules themselves. Below that depth lies the aphotic zone, the realm without light, a volume of water that contains 90% of the ocean's total volume and harbours ecosystems that function on entirely different principles than the sunlit surface waters above. The only illumination that ever appears in these depths comes from bioluminescence, the cold biological fire that some organisms produce through chemical reactions, flashes and glows that serve as lures for prey, signals to potential mates, warnings to predators, or simple camouflage against the faint down-welling light that filters through from above in the upper reaches of the twilight zone. And here, in this alien realm that seems impossibly hostile to any form of life as we understand it, anchored to the dark volcanic rock of the seafloor by threads finer than a strand of spider silk, something waits with the patience of stone. It looks like a miniature tree crafted from translucent glass by some impossibly skilled artisan, each branch terminating in a perfect sphere no larger than a marble, each sphere catching and refracting the LED lights of the remotely operated vehicle in ways that make the whole structure seem to glow with an inner luminescence it does not actually possess. The organism stands perhaps 10 centimeters tall, no bigger than your hand, delicate and ethereal in its construction, appearing so fragile that a strong current might shatter it into 1,000 crystalline fragments. It does not move, and it cannot move, possessing no muscles, no tendons, no joints, no nervous system to coordinate movement even if it had the anatomical machinery to execute such commands. It has no eyes to see prey approaching, no nose to smell chemical signals in the water, no ears to detect vibrations. By all conventional measures, it should be one of the most helpless organisms in the ocean, a sessile creature at the mercy of whatever currents and chance deliver to it. Yet despite this apparent fragility, despite its plant-like stillness, despite its complete lack of any organ we would associate with predation or even basic awareness, this organism is a hunter, as efficient and deadly as any great white shark patrolling the sunlit waters 2 miles above. It is as patient as the most successful spider sitting motionless at the center of its web, waiting for vibrations that signal a trapped fly. It is as perfectly adapted to its specific environment and ecological niche as any organism that has ever evolved on this planet across 4 billion years of life's history. This is Chondrocladia, a member of a remarkable group of animals that has accomplished something that should be impossible according to our traditional understanding of what sponges are and how they function. It has transformed itself from a passive filter feeder into an active carnivore, has turned its body into a trap, has found a way to capture and consume live prey without possessing any of the features we normally associate with predatory life. And in doing so, it has revealed to us that the creativity of evolution, the sheer inventiveness of natural selection working across millions of years, exceeds anything we could have imagined sitting in our comfortable laboratories trying to understand how life works. The discovery of the deathball sponge, as it would come to be called by scientists and science journalists alike, did not happen by accident. It resulted from one of the most ambitious deep-sea expeditions ever mounted to explore the Southern Ocean, that vast ring of cold water that encircles Antarctica and connects the Atlantic, Pacific and Indian Oceans in a continuous band of some of the roughest seas on Earth. In March of 2025, the research vessel Fulcor 2, operated by the Schmidt Ocean Institute, arrived in the waters around the South Sandwich Islands, a remote volcanic archipelago that rises from the Scotia Ridge in the South Atlantic Ocean, roughly 800 kilometers southeast of South Georgia Island. This is one of the most isolated places on the planet, so far from any continental landmass that during the expedition, the crew and scientists aboard joked that the closest other humans were astronauts aboard the International Space Station orbiting overhead. The Falcor is not an ordinary ship. It is a purpose-built oceanographic research vessel equipped with technology that would have seemed like science fiction just a generation ago. Its hull bristles with sophisticated sonar arrays that can map the seafloor in 3 dimensions with resolution measured in centimeters. Its laboratories contain genomics equipment, microscopes, chemical analyzers, aquarium systems for keeping deep-sea organisms alive under pressure, and computer workstations running software that can process terabytes of data in real time. But the ship's most important asset is its remotely operated vehicle, or ROV, named Sebastian after the Schmidt family's love of maritime exploration. This ROV is essentially a robot submarine, an unmanned submersible that can dive to depths of 4,500 meters, equipped with high-definition cameras, powerful LED lighting arrays, multiple robotic manipulator arms that can collect specimens with surgical precision, sensors to measure temperature, salinity, oxygen concentration, pH, and other chemical properties of the water, and a fiber-optic tether that connects it to the ship above, allowing real-time control and data transmission. On the 17th of March, 2025, Sebastian descended through the water column toward the seafloor near one of the South Sandwich volcanoes, passing through the familiar stratified zones of the ocean. The sunlit surface layer, or epipelagic zone, extends from the surface down to about 200 meters, where enough light penetrates to support photosynthesis. Below that lies the twilight zone, or mesopelagic zone, from 200 to 1,000 meters, where sunlight filters down but not enough for plants to grow, where many fish have enormous eyes to catch every available photon, where bioluminescence becomes the dominant form of illumination. Deeper still lies the midnight zone, or bathypelagic zone, from 1,000 to 4,000 meters, where no sunlight penetrates at all, where the pressure climbs toward levels that would crush most surface organisms, where the temperature drops toward freezing, where food becomes increasingly scarce as the distance from the productive surface waters increases. At 3,600 meters, Sebastian reached the abyssal seafloor, that vast plain of sediment and volcanic rock that covers more than half of Earth's surface, yet remains less explored than the surface of Mars. The ROV's lights swept across a landscape that looked simultaneously alien and strangely familiar, a world of mud and rock and occasional outcrops of harder substrate, populated by creatures that seemed to belong to a different evolutionary experiment than the life we know from shallow waters. Sea cucumbers crawled across the sediment, vacuum cleaners of the deep processing mud to extract whatever microscopic organic particles had drifted down from above. Brittle stars, relatives of starfish with impossibly long serpentine arms, clung to rocks and stretched their arms into the current to catch passing food particles. Glass sponges, those ancient animals whose skeletons are made of pure silica, stood like frozen fountains on the seafloor, their bodies built from thousands of interlocking glassy spicules that form lattices of extraordinary beauty and complexity. And then the cameras caught something unexpected. Among the more familiar deep-sea fauna anchored to a volcanic rock outcrop stood a grove of structures unlike anything the scientists aboard the Falcor had seen before. They looked like miniature trees, each no more than 10 or 12 centimeters tall, with a central stalk rising from the rock and multiple branches radiating outward at regular intervals. At the tip of each branch was a translucent sphere, perfectly round, about the size of a marble, that seemed to glow when the ROV's lights hit it from certain angles. The spheres were not glowing by their own bioluminescence, but rather refracting and scattering the artificial light in complex ways that suggested an internal structure of remarkable sophistication. The entire organism, from base to branch tips, appeared to be made of glass, or something very much like glass, a material at once incredibly delicate and surprisingly strong. The pilot manoeuvred Sebastian closer, bringing the ROV's high-definition cameras to within centimeters of one of these strange structures. The scientists watching the video feed from the control room aboard the ship leaned forward in their chairs, trying to make sense of what they were seeing. The central stalk of the organism was about the diameter of a pencil, slightly thicker at the base where it attached to the rock, tapering slightly as it rose. The surface appeared smooth at first glance, but under magnification revealed a complex texture, a landscape of microscopic spicules, tiny needle-like structures made of silica, the same mineral that forms glass and quartz. These spicules were arranged in precise patterns, overlapping and interlocking to create a flexible but strong scaffold that supported the entire structure. The branches radiated from the stalk at regular intervals, typically 4 to 6 branches per node, arranged in a spiral pattern that maximized the organism's exposure to the surrounding water. Each branch was perhaps 2 to 3 centimeters long, tapering from the stalk toward the terminal sphere. And it was these spheres that captured everyone's attention, because under close examination they revealed themselves to be far more complex than simple balloons of tissue. Each sphere was a precisely engineered structure, an inflatable trap that remained turgid through internal water pressure, maintained by a vestigial water canal system, the last remnant of the aquiferous system that all other sponges use for feeding but that carnivorous sponges have largely abandoned in favour of their predatory lifestyle. Dr. Michelle Taylor, the chief scientist aboard the Falkor for this expedition and a marine biologist specialising in deep-sea ecosystems, recognised immediately that they were looking at something significant. The overall morphology suggested a sponge, specifically a member of the family Cladohisidae, the carnivorous sponges, but the arrangement of the terminal spheres was unusual, different from any of the roughly 150 described species in this fascinating family. Using Sebastian's manipulator arms, the team carefully collected several specimens, placing them gently in special containers designed to maintain pressure and temperature during the ascent to the surface. Over the next several hours, as Sebastian explored more of the surrounding seafloor, the team discovered dozens of these sponges, some solitary, others growing in small clusters, all anchored to hard volcanic rock in areas where the current was strong enough to bring a steady supply of potential prey, but not so strong as to tear the delicate structures from their moorings. Back aboard the ship, the specimens were transferred to a specialised pressure aquarium, a high-tech system that can maintain deep-sea organisms at their natural pressure while allowing scientists to observe and study them. Under the controlled conditions of the laboratory, the team began the painstaking work of examining these creatures in detail. What they discovered over the following days and weeks would add a fascinating new chapter to our understanding of how evolution can take a basic body plan and modify it in radical ways to exploit new ecological opportunities. The deathball sponge, as the team began calling it based on the terminal spheres that gave it such a distinctive appearance, is a carnivore, a predator that feeds on small crustaceans and other invertebrates that drift or swim into contact with its branches. Unlike the vast majority of sponges, which are filter feeders that pump water through their bodies to strain out bacteria and other microscopic food particles, carnivorous sponges have evolved an entirely different feeding strategy in response to the unique challenges of life in the nutrient-poor deep sea. They have lost the chonocytes, those specialised flagellated cells that create water currents in normal sponges, and they have lost the complex system of canals and chambers through which water flows. In their place, carnivorous sponges have evolved a hunting strategy based on passive trapping and active digestion. The key to understanding how these sponges hunt lies in examining the fine structure of their surface, particularly the arrangement of the microscopic spicules that cover every part of their body. Sponges produce spicules as part of their skeletal system, tiny structures made of either silica or calcium carbonate that provide structural support and in some cases defense against predators. In most sponges, these spicules are embedded within the tissue or form a loose scaffold. In carnivorous sponges, however, the spicules have been modified into weapons. Under scanning electron microscopy, the surface of a carnivorous sponge reveals a forest of spicules projecting outward from the tissue, each one shaped like a tiny hook or anchor. These are called sigmoncistras in the technical literature, modified versions of a more common spicule type called sigmas that have been bent and twisted into shapes that are perfect for snagging and holding soft-bodied prey. When a copepod or amphipod or other small crustacean touches the sponge's surface, perhaps while swimming past or being carried by a current, these hooked spicules catch on the creature's exoskeleton or appendages. The prey struggles, as prey always does, but every movement drives the hooks deeper, entangling it more thoroughly in a microscopic cage from which there is no escape. What happens next is perhaps even more remarkable than the initial capture. Once the prey is secured, specialised cells within the sponge's tissue begin to move. These are archaeocytes, multipotent cells that are something like stem cells in their ability to differentiate into various cell types and perform various functions. In carnivorous sponges, archaeocytes migrate toward the trapped prey, moving through the mesohyl, that gelatinous middle layer of the sponge's body. When they reach the prey, they begin to secrete digestive enzymes directly onto and into the captured organism. This external digestion breaks down the prey's tissues into smaller molecules that can be absorbed. The process takes hours or even days depending on the size of the prey, and it happens without any of the anatomical structures we normally associate with digestion. There is no mouth, no stomach, no intestine, just cells secreting enzymes and absorbing the resulting nutrients directly through their membranes. It is digestion reduced to its most basic cellular level, a process that harks back to how single-celled organisms consume their food, yet accomplished by a complex multicellular animal that has dispensed with many of the anatomical features we consider essential to animal life. The terminal spheres on the deathball sponge represent an evolutionary innovation specific to the genus Chondrocladia. While many carnivorous sponges have filaments or branches covered with hooks, Chondrocladia has evolved these inflatable structures that maximize surface area for prey capture, while minimizing the amount of tissue that must be maintained. Each sphere is essentially a balloon maintained by internal pressure, its walls thin but reinforced by a network of spicules that prevent it from bursting. The surface of each sphere is covered with the same hook-like spicules found on other parts of the sponge, creating a three-dimensional trap that can capture prey approaching from any direction. But prey capture is only half the story of survival in the deep sea. The other half is reproduction, the ability to create the next generation, to ensure that the species continues even in an environment where individuals may be separated by hundreds of meters of empty seafloor, and where finding a mate presents challenges that would seem insurmountable. Carnivorous sponges, like their filter-feeding relatives, have evolved reproductive strategies that are both bizarre and brilliant, solutions to problems that most sexual organisms never face. Sponges as a group are capable of both sexual and asexual reproduction, and carnivorous sponges use both strategies depending on circumstances. Asexual reproduction is the simpler option, requiring no mate and producing offspring that are genetic clones of the parent. In some carnivorous sponge species, asexual reproduction happens through budding, where a portion of the parent sponge's tissue grows outward, eventually detaching or remaining attached to form a colony of genetically identical individuals. In other species, fragments broken off by currents or physical disturbance can regenerate into complete new individuals, a process called fragmentation that is common throughout the sponge world. The deathball sponges collected by the Falkor expedition showed evidence of both strategies, with some specimens displaying small bud-like outgrowths on their stalks, and others appearing to be clones growing in tight clusters on the same rock. But it is sexual reproduction that reveals just how inventive evolution can be when solving the problem of mating in the deep sea. Most sponges are hermaphrodites, meaning that each individual can produce both sperm and eggs, though usually not at the same time to avoid self-fertilization. The deathball sponge follows this pattern, with different parts of its anatomy dedicated to producing male and female gametes during different reproductive periods. The production of these gametes happens within the mesohyl, that gelatinous middle layer where so much of the sponge's cellular activity takes place. Sperm production in carnivorous sponges is a process unlike that in any other group of animals. Without choanocytes, the flagellated cells that in normal sponges both create feeding currents and can transform into sperm cells, carnivorous sponges must produce sperm from their archaeocytes, those multi-purpose stem-like cells that also handle digestion and tissue repair. The archaeocytes that will become sperm cells gather together in specialized regions of the mesohyl and begin dividing, transforming through a series of stages from large, amoeboid cells into small, streamlined sperm cells equipped with a single flagellum for swimming. What makes this process particularly unusual is that the developing sperm cells are surrounded by a follicle, a thin envelope of tissue formed by a single specialized cell that wraps around the entire cluster of developing sperm. This follicle provides structural support and may also regulate the development process by controlling what nutrients and chemical signals reach the developing sperm cells. As the sperm mature, they remain packed tightly together inside the follicle, eventually forming what looks like a dense ball of cells. When the sperm are fully developed, the follicle ruptures, releasing the sperm into the surrounding tissue. But releasing sperm into your own tissue solves nothing if the goal is to find an egg in a different individual. How does sperm get from one sponge to another when individuals may be separated by meters or tens of meters of empty water, when there are no breeding migrations, no courtship displays, no deliberate search for a mate? The answer lies in a strategy that seems almost impossibly risky, but apparently works well enough to have persisted for millions of years. The mature sperm are packaged together into spermatic cysts, essentially bundles of thousands or tens of thousands of sperm cells held together by a thin membrane. These cysts are released from the parent sponge into the surrounding water, where they drift with the current. This is a strategy of spectacular inefficiency. Most of these sperm bundles will drift away into the darkness, encountering nothing, their genetic cargo lost forever. But some, a tiny fraction, will drift into contact with another sponge of the same species, and when they do, something remarkable happens. The sponge that receives the sperm bundle does not simply absorb it. Instead, the sperm must work their way through the tissue of the receiving sponge, swimming through the mesohyl toward the eggs that are developing in a different part of the same sponge. This internal fertilization, happening within the body of what is technically a second individual, but which serves as the site of fertilization rather than simply as an egg provider, is unique to sponges and represents yet another example of how these ancient animals have evolved solutions to reproductive challenges that differ fundamentally from what we see in other animal phyla. Once fertilized, the eggs develop within the parent sponge's tissue for a period that varies by species but can extend for weeks or even months. The developing embryos are nourished by nutrients from the parent, receiving a steady supply of the organic molecules that the parent has extracted from captured prey. This extended brooding period is common in deep-sea organisms, which tend to invest heavily in each offspring rather than producing huge numbers of small, poorly provisioned young as many shallow water species do. In an environment where food is scarce and predation pressure is high, quality matters more than quantity. The embryos eventually develop into larvae, small free-swimming stages that represent the dispersal phase of the life cycle. Sponge larvae are among the most unusual larvae in the animal kingdom, tiny elongated creatures covered with cilia, those hair-like projections that beat in coordinated waves to propel the larva through the water. The larvae of carnivorous sponges are particularly interesting because they retain some features that adult carnivorous sponges have lost. They have a simple aquiferous system with chonocyte chambers, suggesting that the larva may actually filter feed during its brief free-swimming period, supplementing the energy stores it received from its parent with fresh nutrients captured from the water. This larval stage is crucial for dispersal. The adults are sessile, permanently anchored to the rock where they settled, unable to move even if conditions deteriorate or competition becomes intense. The larvae, swimming weakly but persistently through the water column, can travel meters or even kilometers from their point of origin before settling, carried by currents that flow across the seafloor in patterns we're only beginning to understand. This dispersal capability is what allows carnivorous sponges to colonize new areas, to maintain gene flow between populations that might otherwise become isolated, to persist as a species even though any single population may be wiped out by a volcanic eruption, a seafloor landslide, or some other catastrophe. When a larva finds suitable habitat, something that may take hours or days or even weeks depending on local conditions and luck, it settles onto a hard surface and begins its transformation into an adult. The process is called metamorphosis, and it involves a complete reorganization of the larva's body plan. The cilia are lost. The aquiferous system is largely dismantled. The body elongates, forming the stalk that will anchor the sponge for the rest of its life. Specialized cells begin producing spicules, building the skeleton that will support the growing sponge and provide the hooks needed for prey capture. And archaeocytes, those multi-purpose cells that do so much of the real work in sponge biology, begin migrating through the developing tissue, setting up the zones that will become branches, that will produce gametes, that will handle digestion when the first prey is captured. The terminal spheres that give the deathball sponge its distinctive appearance develop relatively late in this process, emerging as outgrowths from the tips of the branches that themselves grew from the central stalk. Each sphere begins as a small bump, a local thickening of tissue that gradually expands, becoming hollow as internal water pressure pushes outward against the thin walls. The expansion is controlled and gradual, proceeding over weeks or months as the young sponge grows toward its adult size. The spicules that will cover the surface of each sphere are produced by sclerocytes, specialized cells that take up dissolved silica from the surrounding seawater and use it to build these remarkable structures. The process of spicule formation is itself a minor miracle of cellular engineering. Silica, or silicon dioxide, is the second most abundant mineral in Earth's crust after oxygen. It is the main component of sand, quartz, and glass. In seawater, silica is dissolved at low concentrations, typically between 1 and 100 micromolar depending on depth and location. Sponges have evolved specialized enzymes called silicatins that can capture dissolved silica and precipitate it as solid silicon dioxide with extraordinary precision. The sclerocyte begins by secreting a proteinaceous scaffold, an organic framework that serves as a template for silica deposition. The silicatin enzymes then catalyze the polymerization of silica onto this template, building up the spicule layer by layer, atom by atom, until it reaches its final size and shape. The spicules in carnivorous sponges come in various forms, each serving a specific function. Megascleres are the large spicules that form the main structural skeleton, giving the sponge its shape and strength. In the death ball sponge, these are primarily monaxons, rod-like spicules that can be straight or slightly curved, measuring anywhere from a few hundred micrometers to several millimeters in length. They are arranged in bundles that run longitudinally through the stalk and radiate outward through the branches, creating a flexible but strong scaffold that can bend with currents without breaking. Microscleres are smaller spicules that serve various functions, including defense and prey capture. The sigmoid sistras that cover the surface of the spheres and branches are microscleres, each one precisely shaped into a hook that can snag prey. What makes sponge spicules particularly remarkable is their material properties. They are made of hydrated amorphous silica, essentially biological glass, yet they exhibit mechanical properties that far exceed those of manufactured glass. Where human-made glass is brittle, shattering catastrophically when stressed beyond its breaking point, sponge spicules are remarkably tough, able to bend and flex without fracturing. This toughness comes from their internal structure. Spicules are not solid rods of glass, but rather composite structures, consisting of alternating layers of silica and protein. The proteins, trapped within the silica matrix during formation, create weak interfaces that can deflect propagating cracks, preventing the catastrophic failure that would occur in pure silica. Some species of glass sponges produce spicules that are truly extraordinary in their dimensions. Monohaphis chuni, a deep-sea species found in the Pacific Ocean, produces basal spicules that can reach 3 meters in length and 1 centimeter in diameter, the largest known biological structures made of silica. These giant spicules anchor the sponge to the soft sediment of the abyssal seafloor, functioning like tent stakes driven deep into the mud. They are constructed from hundreds of concentric layers of silica, each layer about 5 to 10 micrometers thick, deposited sequentially over what must be years or even decades of continuous growth. The resulting structure has remarkable optical properties, functioning as a biological fiber optic cable that can transmit light along its entire length with minimal loss. While the deathball sponge does not produce spicules of such gigantic proportions, its spicules are nonetheless sophisticated structures adapted to the specific mechanical and functional demands of its lifestyle. The stalk spicules must provide vertical support, resisting compression forces as well as bending moments imposed by currents. The branch spicules must be strong enough to support the terminal spheres while remaining flexible enough to move with the flow, and the surface spicules must be sharp and strong enough to penetrate the exoskeletons of small crustaceans without breaking, functioning as biological fishhooks that must work reliably every single time. The ecosystem in which the deathball sponge lives is shaped by forces that have no parallel in shallow water. The deep sea is not a homogeneous environment, but rather a patchwork of different habitats and energy sources, each supporting its own community of specialized organisms. In most of the deep ocean, life depends ultimately on organic matter that sinks from the sunlit surface waters above, a process called the biological pump that transfers carbon and other nutrients from the productive surface to the food-starved abyss. Dead phytoplankton, fecal pellets from zooplankton, aggregates of mucus and organic particles called marine snow, even the occasional carcass of a fish or marine mammal, all sink through the water column at rates determined by their size and density, providing a slow but steady rain of food to the deep sea floor. But this rain of organic matter is far less abundant than the food available in surface waters. By the time particles reach the abyssal seafloor 3 or 4 kilometers down, 95% or more of the organic carbon that was produced at the surface has already been consumed or decomposed by bacteria and animals in the overlying water column. What finally reaches the bottom is mostly refractory material, organic compounds that resist bacterial decomposition mixed with the mineral shells and skeletons of surface-dwelling organisms. The result is that food availability in the deep sea is typically 100 to 1,000 times lower than in shallow coastal waters, measured as grams of organic carbon per square metre of seafloor per year. This extreme scarcity of food has profound consequences for deep-sea ecology. Population densities are low because there simply is not enough energy to support large numbers of individuals. Organisms tend to be small, conserving energy and requiring less food. Metabolic rates are reduced, with many deep-sea animals having metabolisms that run at half or even a quarter of the rate of their shallow water relatives. Growth is slow, taking years or decades to reach adult size. Reproduction is infrequent, perhaps occurring only once in an individual's lifetime or at intervals of several years. And competition for food is intense when food does appear, with scavengers like amphipods and isopods swarming to carcasses within hours of their arrival on the seafloor, stripping them to bare bone in a matter of days. In this context, the evolution of carnivory in sponges makes perfect evolutionary sense. Filter feeding, the ancestral feeding mode for sponges, works well when bacteria and small particles are abundant in the water. A sponge pumps water through its body at rates that can reach hundreds of litres per day, straining out bacteria with efficiency that approaches 90%. But this strategy requires energy, the energy needed to power the beating flagella of millions of chonocyte cells working continuously to create the water current. And it requires that the water being pumped contains enough bacteria to make the effort worthwhile. In the deep sea, bacterial concentrations are typically 10 to 100 times lower than in shallow water, measured as cells per milliliter. Pumping large volumes of nearly bacteria-free water through your body makes no thermodynamic sense. The energy cost of pumping exceeds the energy gained from the scarce bacteria you manage to capture. Better to shut down the pumping system entirely, reduce your metabolic rate to the bare minimum needed to stay alive, and wait for something more substantial to come along. And if you can evolve structures to capture that more substantial something when it does arrive, so much the better. This is exactly what carnivorous sponges have done. They have abandoned the energetically expensive business of filter feeding and instead invested in structures, the hooked spicules, that can capture larger, more nutritious prey with no ongoing energy cost. The hooks are always there, always ready, requiring no metabolic energy to maintain once they have been produced. When prey arrives, randomly, carried by currents or through its own swimming efforts, the hooks do their job automatically, mechanically, with no need for neural control or muscular effort. Only after capture, when enzymes are being secreted and nutrients are being absorbed, does the sponge invest significant energy in the feeding process. It is an energy budget strategy perfectly suited to an environment where food is rare but nutritious when it appears, where the optimal strategy is to minimize baseline metabolic costs and respond opportunistically when resources become available. But the deep sea around the South Sandwich Islands, where the deathball sponge was discovered, is not entirely dependent on the rain of organic matter from surface waters. This region is volcanically active, part of the Scotia Arc, a chain of submarine and island volcanoes that marks the boundary between the South American and Antarctic tectonic plates. Where plates meet and magma rises toward the surface, the seafloor can be punctured by hydrothermal vents, those remarkable hot springs where mineral-rich fluid heated by contact with magma erupts from cracks and chimneys on the ocean floor. Hydrothermal vents create oases of life in the deep sea, ecosystems that do not depend on photosynthesis, but instead derive their energy from chemosynthesis, the process by which certain bacteria harness the chemical energy stored in reduced compounds like hydrogen sulfide, methane, and hydrogen gas. These chemicals are abundant in vent fluids because the extreme heat and pressure of the subsurface hydrothermal system create reducing conditions that transform the oxidized minerals in rock into reduced forms that are thermodynamically unstable when exposed to the oxygen-rich seawater of the deep ocean. When vent fluid mixes with seawater, the reduced chemicals react with oxygen, releasing energy that chemosynthetic bacteria can capture and use to fix carbon dioxide into organic compounds through various metabolic pathways. The most studied of these chemosynthetic pathways is sulfide oxidation, used by bacteria that obtain energy by oxidizing hydrogen sulfide to elemental sulfur or sulfate. The simplified reaction can be written as hydrogen sulfide plus oxygen, yields elemental sulfur plus water plus energy, though the actual biochemistry involves multiple steps and intermediate compounds. The energy released by this reaction is captured in the form of ATP, the universal energy currency of cells, which the bacteria then use to power the Calvin cycle or other carbon fixation pathways that convert carbon dioxide into organic molecules like sugars. These chemosynthetic bacteria form the base of hydrothermal vent food webs, occupying the same ecological role that photosynthetic phytoplankton play in sunlit surface waters. Some vent bacteria live freely in the water or form mats on rock surfaces. Others have entered into intimate symbiotic relationships with animals, living inside specialized organs where the host provides them with hydrogen sulfide and oxygen, while they provide the host with organic compounds synthesized through chemosynthesis. The giant tube worms of the genus Riftia, which can reach 2 meters in length and are among the most conspicuous animals at vent sites, have no mouth, no gut, no ability to feed conventionally. Instead, they harbor billions of sulfide-oxidizing bacteria in a specialized organ called a trophosome, and they derive all their nutrition from organic compounds synthesized by these bacterial partners. The productivity of hydrothermal vent ecosystems is extraordinary compared to the surrounding deep-sea floor. Estimates suggest that chemosynthetic bacteria at vents can produce more than 4,000 tons of organic carbon per day worldwide, a staggering amount considering the relatively small total area occupied by vent habitats. On a per-volume basis, vent ecosystems are among the most productive in the ocean, comparable to tropical rainforests or coral reefs in terms of the biomass they support. This high productivity supports dense populations of animals including shrimp, crabs, limpets, tube worms, mussels, and many others, most of them species found nowhere else, highly specialized for life in the unique conditions of hydrothermal vent environments. Carnivorous sponges, including the deathball sponge, are found both in vent habitats and in the surrounding deep sea far from any vent influence. This suggests that they are eurotopic, able to thrive across a range of environmental conditions and food availability levels, taking advantage of whatever prey happens to be present rather than specializing on a specific food source. In the vicinity of vents, where animal populations are dense and productivity is high, carnivorous sponges may capture more prey and grow faster than their counterparts in vent-free areas. But even in these productive environments, they must compete with many other predators for the available prey, including fish, crabs, and other invertebrates that can move actively through the environment searching for food. The food web connections at hydrothermal vents are complex and have been the subject of intensive research since vents were first discovered in 1977. At the base are the chemosynthetic bacteria, some free-living and some living in symbiotic association with animals. These bacteria are consumed by various herbivores, which is perhaps a misnomer since they are eating bacteria rather than plants, but the term captures the ecological role of primary consumers that feed on primary producers. Copepods, amphipods, and other small crustaceans graze on bacterial mats or filter bacteria from the water. Protists, single-celled eukaryotes that function as the ocean's main microbial predators, consume bacteria at astonishing rates. Studies showing that protists can consume between 28 and 62% of the daily bacterial and archaeal biomass production in hydrothermal vent fluids. These primary consumers are in turn eaten by secondary consumers, including larger crustaceans, fish, and carnivorous sponges. And those secondary consumers are eaten by tertiary consumers in a food web that can extend to 4 or 5 trophic levels in some vent systems. Energy is lost at each transfer from one trophic level to the next. Typically around 90% of the energy is lost to metabolism and is not available to the next trophic level, which limits how many levels a food web can support. But at vents, where the input of energy from chemosynthesis can be very high, food webs can be more complex and support higher biomass than would be possible based on the rain of organic matter from surface waters alone. Carnivorous sponges fit into this web as specialized predators of small mobile invertebrates, particularly crustaceans. Isotope studies, which measure the ratio of stable isotopes of carbon and nitrogen in an organism's tissues to determine what it has been eating, have confirmed that carnivorous sponges feed primarily on animals rather than bacteria or detritus. The nitrogen isotope ratios in their tissues are elevated compared to filter-feeding sponges and herbivorous invertebrates, placing them clearly in the role of secondary or tertiary consumers. Some species show isotope signatures suggesting they feed on animals that themselves fed on chemosynthetic bacteria, connecting them directly to vent-based food webs, even when the sponges themselves are located some distance from active venting. The role of carnivorous sponges in deep-sea ecosystems is not fully understood, partly because they were only discovered relatively recently, and partly because studying them in their natural habitat is challenging and expensive. But they are likely more important than their modest size and sedentary lifestyle might suggest. By capturing and consuming mobile crustaceans, they recycle nutrients that might otherwise disperse widely across the seafloor or be transported away from the area entirely. They provide food for their own predators, which may include sea stars and fish capable of consuming them despite the deterrent effect of their spicules. And they may compete with other predators for the same prey, influencing community structure and food web dynamics in ways that are difficult to predict without detailed ecological studies. The bacterial communities associated with carnivorous sponges add another layer of complexity to their ecology and biochemistry. All sponges harbour dense and diverse communities of microorganisms, mostly bacteria, but also archaea and fungi, living within the mesohyl and sometimes within specialised cells. These sponge-associated microbes can constitute up to 40% of the sponge's total volume in some species, making the sponge as much a mobile microbial habitat as an individual organism. The relationship between sponges and their microbial communities is ancient, probably dating back hundreds of millions of years to the early evolution of sponges, and it has resulted in intricate co-evolutionary partnerships where both parties benefit. In carnivorous sponges, the bacterial community appears to be different from that in filter-feeding sponges, reflecting the different nutritional resources available. Filter-feeding sponges process large volumes of seawater containing bacteria, organic particles, and dissolved organic matter, providing their symbiotic bacteria with a continuous supply of diverse nutrients. Carnivorous sponges, by contrast, feed episodically on large prey items that provide a sudden pulse of protein, amino acids, and other nitrogen-rich compounds when digested. This difference in nutrient supply appears to select for different bacterial communities, with carnivorous sponges harbouring higher abundances of bacteria capable of nitrogen cycling and amino acid metabolism. The bacterial community in a carnivorous sponge is not randomly assembled, but rather represents a selected consortium of organisms that can thrive in the specific conditions within the sponge's tissues and provide services valuable to the host. Some bacteria may help with prey digestion, secreting enzymes that complement those produced by the sponge's own cells, breaking down complex molecules into forms that can be more easily absorbed. Others may process waste products from protein metabolism, particularly ammonia, which is toxic at high concentrations and must be converted to less harmful forms or excreted. Still others may synthesize vitamins, amino acids, or other essential compounds that the sponge cannot make itself, or may produce defensive chemicals that deter predators or competitors. Different parts of the sponge's body harbour different bacterial communities, suggesting that these communities are spatially structured according to the local conditions and requirements of different tissues. The stalk may have one assemblage of bacteria adapted to structural support and waste processing. The branches may have another assemblage involved in nutrient distribution and spicule formation. And the terminal spheres, those remarkable inflatable traps, may have yet another community specialised for the intense burst of metabolic activity that follows prey capture when digestive enzymes are being secreted at high rates and nutrients are flooding into the tissue. This concept of the sponge as a holobionт, an ecological unit consisting of the host animal plus its associated microbial community, has become increasingly important in marine biology over the past 2 decades. It recognises that we cannot fully understand an organism by studying only its genes and cells and anatomy. We must also understand the microbes it harbours, their metabolic capabilities, their interactions with each other and with the host, and how the entire assemblage responds to environmental changes and stressors. For carnivorous sponges living in the extreme environment of the Antarctic deep sea, this holobionт perspective is particularly important because many of the adaptations that allow them to survive may be microbial rather than inherent properties of the sponge itself. The Antarctic connection to carnivorous sponges runs deeper than simply being one of many places where these organisms are found. The Southern Ocean and its deep-sea environments may have played a crucial role in the evolution and diversification of these remarkable predators, creating conditions that both favoured the evolution of carnivory and allowed carnivorous lineages to diversify into the many species we see today. The Antarctic deep sea represents one of the most extreme environments these creatures inhabit, combining crushing pressure, frigid temperature, extended or complete isolation from surface productivity, and a degree of environmental stability that is unusual even for the generally stable deep ocean. The Southern Ocean is geographically isolated from other ocean basins by the Antarctic Circumpolar Current, the ACC, a massive current system that flows endlessly eastward around Antarctica, driven by the fierce westerly winds that circle the southern high latitudes in a band meteorologists call the Roaring Forties, Furious 50s, and Screaming 60s based on their latitude. This current is the largest on Earth by volume transport, moving approximately 150 million cubic meters of water per second eastward, a flow rate that exceeds the combined flow of all the world's rivers by a factor of several hundred. The current extends from the surface all the way to the seafloor in many areas, and its eastward flow creates an effective barrier to north-south dispersal for many marine organisms, particularly those with limited swimming ability or short-lived larvae that would be swept downstream before they could cross from one side to the other. This isolation has been in place for millions of years, since the Antarctic Circumpolar Current fully developed around 34 million years ago, when the Drake Passage finally opened completely between South America and the Antarctic Peninsula, allowing unrestricted flow around the continent. The result is that Antarctic marine fauna has evolved partly in isolation from temperate and tropical faunas, developing endemic species found nowhere else, while retaining some ancient lineages that went extinct elsewhere. The deep sea around Antarctica contains a mix of cosmopolitan species found throughout the world's oceans, species that probably dispersed when Antarctica was connected to other continents, or when ocean circulation patterns were different, and endemic species that evolved within the Southern Ocean and have never spread beyond it. The Antarctic bottom water, ABW, the coldest and densest water mass in the world's oceans, forms primarily in a few specific locations around Antarctica, where conditions allow extremely cold, salty water to sink from the surface to the abyssal depths. The primary formation sites are the Weddell Sea, the Ross Sea, the Adélie Coast, and near Cape Darnley, regions where polynyas, areas of open water within the winter sea ice, allow intense heat loss from the ocean to the atmosphere during the Antarctic winter. As sea ice forms at the surface, salt is excluded from the ice crystals in a process called brine rejection, making the water, beneath the ice, even saltier than normal. This cold, salty water becomes extremely dense, denser than any other water in the ocean, and it sinks, flowing down the continental slope under the force of gravity, spreading out across the abyssal plains and eventually filling the deepest parts of all the ocean basins that have connections to the Southern Ocean. Temperatures in Antarctic bottom water can drop as low as -2.3 degrees Celsius, far below the normal freezing point of pure water, which is 0 degrees Celsius, kept liquid only by the salt content, which lowers the freezing point, and by the immense pressure, which also depresses the freezing point by approximately 0.07 degrees per 100 meters of depth. At 3,000 meters, the pressure depression of the freezing point is about 2 degrees, meaning that water can remain liquid at -2 degrees even with the salt content. This is close to the absolute minimum temperature that liquid water can achieve on Earth under natural conditions, and many organisms simply cannot survive in such cold water, because ice crystals form in their tissues despite the salt and pressure, rupturing cells and causing death. But carnivorous sponges thrive in this water, living their entire lives at temperatures that would kill most other organisms within hours. They have evolved biochemical adaptations to prevent ice formation, possibly including antifreeze proteins similar to those found in Antarctic fish, compounds that interfere with ice crystal growth by binding to the crystal surfaces and preventing water molecules from adding to the structure. They have enzymes that continue to function at temperatures where most enzymes would become too rigid to catalyze reactions efficiently. They have membranes that remain fluid despite the cold, incorporating unsaturated lipids that have lower freezing points than the saturated fats that predominate in warm water organisms. Every aspect of their cellular biochemistry has been fine-tuned by selection to operate in this extreme environment. The question of how carnivorous sponges first evolved has intrigued biologists since these remarkable animals were first discovered. The transition from filter feeding to carnivory represents a fundamental change in lifestyle, requiring not just morphological modifications but also behavioral and physiological adaptations. How does a sessile animal, that has spent hundreds of millions of years perfecting the art of pumping water and filtering out bacteria suddenly become a predator? What were the intermediate stages, and what selective pressures drove this evolutionary transition? Molecular phylogenetic studies, which use DNA sequence comparisons to reconstruct evolutionary relationships, have shown that all known carnivorous sponges form a monophyletic group, meaning they all share a common ancestor, and that ancestor was the first carnivorous sponge. This is important because it tells us that carnivory evolved only once in sponges, rather than multiple times independently in different lineages. Everything we see in modern carnivorous sponges, all 150 or more described species spanning multiple genera and displaying a remarkable diversity of body forms, descended from that single ancestral species that first made the transition from filter feeding to predation. When did this happen? We do not know precisely because the fossil record of sponges is frustratingly incomplete. Sponges have soft bodies that do not preserve well, and only their spicules typically survive as fossils. While these spicules can sometimes be diagnostic enough to identify the family or even genus of sponge that produced them, they cannot always tell us what feeding mode that sponge used. Carnivorous sponges have distinctive hooked spicules, but similar shapes can occur in some filter-feeding sponges that use them for other purposes, making it difficult to identify carnivorous species from spicules alone without the context of the whole animal. However, we can make some educated guesses based on molecular clock analyses, which use the rate of DNA sequence evolution to estimate when different lineages diverge from common ancestors. These analyses suggest that the family Cladohisidae, containing all carnivorous sponges, diverged from their filter-feeding relatives sometime in the Mesozoic era, perhaps around 150 to 200 million years ago during the Jurassic or Cretaceous periods. This was a time of major changes in ocean chemistry and circulation, a time when many modern marine groups were diversifying and establishing the ecological relationships we see today. It was also a time when the deep sea was becoming more isolated from surface processes as ocean stratification increased, potentially creating the conditions that favoured the evolution of specialised deep-sea predators. The selective pressure that drove the evolution of carnivory in sponges was almost certainly food limitation in the deep sea. As sponges colonised deeper and deeper waters, following the general trend of marine life expanding into all available habitats, they encountered environments where filter feeding became progressively less effective due to declining bacterial concentrations. At some threshold depth, probably somewhere in the lower bathyal or abyssal zones, the energy cost of pumping water exceeded the energy gained from captured bacteria, making filter feeding thermodynamically impossible. Sponges that found themselves in these environments faced a stark choice: either evolve a different feeding mode or go extinct. The ancestors of modern carnivorous sponges must have possessed some pre-adaptation that made the transition to carnivory possible. Perhaps they already had hooked spicules used for some other purpose, providing the raw material that could be co-opted for prey capture. Perhaps they had elevated levels of digestive enzymes or particularly active archaeocytes capable of moving to sites of tissue damage or particle aggregation. Whatever the specific pre-adaptations were, they allowed an initial tentative shift toward capturing larger food items, perhaps beginning with dead or dying animals that came to rest on the sponge's surface, rather than active predation on healthy mobile prey. Once this initial shift occurred, natural selection would have favoured elaboration and refinement of the structures and processes involved in carnivory. Individuals with better developed hooks would capture more prey. Individuals with more efficient digestive systems would extract more nutrients from captured prey. Individuals that could survive on less food during the inevitable periods between prey captures would outlast their competitors. Over time, measured in millions of generations and millions of years, these selective pressures would have produced the remarkably specialised carnivorous sponges we see today. Animals that have lost almost all traces of their filter-feeding ancestry and are perfectly adapted to their role as sedentary predators of the deep. The diversification of carnivorous sponges from their common ancestor into the many species we see today represents an equally fascinating evolutionary story. Starting from that first carnivorous species, the lineage radiated into multiple morphological forms, each adapted to slightly different environmental conditions or prey types. Some species evolved tall, branching forms that project high into the water column, maximizing their exposure to currents and potential prey. Others remained small and low, perhaps specializing on prey that lives close to the seafloor. Some developed elaborate branching patterns with hundreds of filaments, Others evolved the balloon-like terminal spheres seen in Chondrocladia. This morphological diversity reflects the creative power of natural selection, working on variation generated by mutation, recombination, and developmental plasticity. The deathball sponge discovered on the expedition to the South Sandwich Islands may represent a new species, though formal description awaits detailed comparison with all previously described species of Chondrocladia and genetic sequencing to determine its exact phylogenetic position. What is clear is that it represents yet another example of the remarkable evolutionary flexibility of this group, another experiment in how to build an effective sedentary predator from the basic sponge body plan. The terminal spheres are not unique to this species but rather characteristic of the genus, yet their specific arrangement, size, and number varies among species and may be adapted to local conditions like current speed, prey availability, or competition from other predators. As we learn more about carnivorous sponges through continued exploration and study, we are also learning about the deep sea itself, about its ecology and evolution, and the forces that shape life in Earth's largest and least known ecosystem. Every new species discovered adds a piece to the puzzle, helping us understand how communities assemble, how energy flows through food webs, how organisms adapt to extreme conditions, and how evolution can produce solutions to ecological challenges that we would never have imagined without seeing the organisms themselves. The future of deep-sea exploration holds immense promise for discovering yet more species of carnivorous sponges and for understanding their biology in greater detail. Advances in remotely operated vehicle technology are making it possible to explore deeper and more remote parts of the ocean with increasing precision and sophistication. New imaging techniques, including 3D reconstruction and molecular imaging, are allowing us to visualize the internal structure and function of these organisms in ways that were impossible just a few years ago. And genomic approaches are revealing the genetic basis of their remarkable adaptations, identifying the genes that allow them to survive at extreme pressure and temperature, that encode their specialized digestive enzymes, that control the formation of their elaborate body structures. The study of carnivorous sponges also has practical implications beyond pure scientific curiosity. The enzymes they use for digestion, capable of breaking down complex proteins and other biomolecules at near-freezing temperatures and high pressure, may have applications in industrial processes or biotechnology. The silicatins they use to construct their spicules represent a form of biomineralization that differs from anything in our current material science toolkit, potentially inspiring new approaches to manufacturing silica-based materials with superior properties. The antifreeze proteins that allow them to function at temperatures below the normal freezing point of water could have applications in food preservation, organ storage for transplantation, or even in developing crops that can tolerate colder climates. More broadly, carnivorous sponges remind us that evolution is more creative than our imaginations, that life can find solutions to problems we would never think to solve in those particular ways, that the natural world still holds countless surprises waiting to be discovered if we have the curiosity and determination to look for them. They challenge our assumptions about what animals can be and how they can live. They expand our understanding of the possible, showing us that a sessile animal without a brain or muscles or specialized sense organs can nonetheless be an effective predator, that an ecosystem based on chemosynthesis rather than photosynthesis can be as productive and diverse as a tropical forest, that life can thrive in conditions that seem impossibly hostile by our surface-dwelling standards. The deathball sponges growing on volcanic rocks 3,600 meters beneath the surface of the Southern Ocean have been there for years or decades or perhaps even centuries. We do not know their lifespan because no one has yet conducted the long-term studies needed to answer that question. They have been catching their microscopic prey and growing slowly in the frigid darkness, reproducing when conditions are right, dispersing their larvae into the black water, hoping that some will find suitable habitats somewhere in the vastness of the abyss. They are part of an ecosystem that operates according to rules, different from those we're familiar with on land or in shallow water, an ecosystem built on chemical energy rather than sunlight, on opportunistic capture of rare prey rather than continuous filter feeding, on slow growth and long lifespans rather than rapid reproduction. And they are part of a planet that remains largely unexplored and poorly understood despite all our technological sophistication. We have mapped the surface of Mars and the Moon in far greater detail than we have mapped the seafloor of our own planet. We have sent spacecraft to the outer solar system and rovers to Mars, yet we have explored less than 20% of the ocean floor with remotely operated vehicles capable of making the detailed observations needed to understand deep-sea ecosystems. The deep sea represents the last great frontier on Earth, a realm covering more than half the planet's surface, yet remaining almost entirely unknown. Every expedition to the deep sea, like the Falkor voyage to the South Sandwich Islands, brings back new species, new observations, new data that change our understanding of how the ocean works and what lives within it. The deathball sponge is just one of dozens of new species discovered during that single expedition, and that expedition is just one of hundreds that have been conducted over the past few decades as deep-sea exploration has accelerated. If every expedition finds this many new species, and if we have explored such a tiny fraction of the deep ocean, then the total biodiversity of the deep sea must be staggering, numbering in the millions of species, most of which science has never seen, and many of which will have body plans and lifestyles as surprising as those of carnivorous sponges. This is both humbling and exciting. Humbling because it reminds us how little we actually know about our own planet, how much remains to be discovered even in the fundamentals of what species exist and where they live and how they make their living. Exciting because it means the age of exploration and discovery is far from over, that future generations will have opportunities to make discoveries as significant as those being made today, that the sense of wonder we feel when encountering something completely new and unexpected can continue indefinitely into the future. The deathball sponge, hanging motionless in the darkness beneath the Antarctic waves, does not know it is remarkable. It does not ponder the evolutionary journey that produced it, or consider how unusual it is to be a predatory sponge rather than a filter feeder. It simply exists, following the ancient biological imperatives encoded in its DNA, capturing prey when opportunity allows, growing slowly, reproducing when ready, maintaining the delicate metabolic balance that allows life to persist in one of the most challenging environments on Earth. But for us, encountering this creature through the cameras of a remotely operated vehicle, operated from a ship on the stormy Southern Ocean, it represents something profound. It represents the extraordinary creativity of evolution, the power of natural selection to produce adaptations we would never have predicted, solutions to problems we might not have recognized as problems. It represents the richness and diversity of life on Earth, the fact that 4 billion years of evolution has produced not one way of being an animal, but countless ways, each perfectly suited to its particular environment and ecological niche. And it represents the possibility of future discovery, the knowledge that somewhere in the unexplored depths, something even more unusual may be waiting, something that will force us once again to reconsider what we thought we understood about how life works. The continued study of carnivorous sponges and other deep-sea organisms is essential not just for satisfying scientific curiosity, but for understanding and protecting Earth's ecosystems in an era of rapid environmental change. The deep sea is not as isolated from human influence as we once thought. Bottom trawling destroys seamount communities. Deep-sea mining threatens to extract mineral resources from the seafloor with unknown consequences for the organisms that live there. Climate change is altering ocean temperatures, circulation patterns, oxygen levels, and chemistry, with effects that cascade through food webs in ways we're only beginning to understand. Plastic pollution has reached the deepest trenches. Chemical pollutants accumulate in deep-sea organisms despite their remoteness from industrial sources. If we're to make informed decisions about how to manage human activities that affect the deep sea, we need to understand what lives there, how ecosystems function, what services they provide, and how they respond to disturbance. This requires baseline data on species diversity and distribution, ecological studies of food webs and community dynamics, physiological research on how organisms cope with extreme conditions, and long-term monitoring to detect changes over time. The discovery of new species like the deathball sponge provides some of this baseline data, establishing what exists in particular locations at particular times, creating a reference point against which future changes can be measured. The story of the deathball sponge is, in the end, a story about the ocean itself, about its vastness and complexity, and the way it continues to surprise us despite centuries of study. It is a story about life's remarkable ability to adapt and persist, to find ways of making a living in environments that seem impossibly hostile, to turn constraints into opportunities. It is a story about evolution, about how the gradual accumulation of small changes over immense spans of time can produce organisms that seem impossibly specialized, yet are perfectly ordinary products of natural processes. And it is a story about human curiosity, about our drive to explore and understand, to push the boundaries of knowledge, to see what lies beyond the next ridge or beneath the next wave. 3,600 meters beneath the surface of the Southern Ocean, in darkness illuminated only by the occasional flash of bioluminescence or the artificial lights of visiting robots, the death ball sponges continue their patient hunt. They have no awareness of the scientists studying them, no knowledge of the excitement their discovery has generated, no concept of being remarkable or unusual. They simply are, existing as they have existed for untold millennia, capturing their microscopic prey, processing the nutrients, growing infinitesimally slowly, releasing their larvae into the current, part of an ecosystem that functions according to ancient rules, in a place that remains alien and mysterious to surface-dwelling creatures like ourselves. And in their simple, strange existence, they embody something essential about life on Earth, something about its diversity and adaptability, and endless capacity to surprise us with solutions we never imagined to problems we barely understood. The timescales on which these sponges operate are almost incomprehensible to organisms like ourselves, whose lives are measured in decades rather than centuries. We do not know how old an individual death ball sponge can become, because no one has yet undertaken the multi-decade monitoring studies that would be required to answer this question. But based on growth rate estimates from other carnivorous sponge species, and from the slow metabolic rates characteristic of Antarctic marine life, it is entirely possible that some of the larger specimens collected by the Falkor expedition are older than the United States as a nation, older than the scientific revolution, perhaps even older than some of the world's great religions. They may have been anchored to their volcanic rock since before Europeans discovered the Americas, growing slowly year after year, century after century, in a place where seasons have no meaning and where time itself seems to have a different quality than it does at the surface. This is the reality of deep-sea life, and of Antarctic life in particular. Growth is slow because metabolism is slow because temperature is cold and food is scarce. Antarctic fish grow at rates 1/10 those of their temperate relatives. Antarctic sea stars may take a decade or more to reach maturity. Some Antarctic sponges, not carnivorous species but filter feeders, have been estimated to be several 1,000 years old based on their size and the known growth rates of similar species in slightly warmer waters. The oldest known individual organism on Earth is not a redwood tree or a bristlecone pine, but possibly a glass sponge from Antarctic waters, though conclusive aging has not been achieved because sponges do not produce annual rings or other structures that allow counting of years. The extreme longevity of Antarctic organisms has profound implications for how their ecosystems function and how they respond to disturbance. In shallow tropical waters, where fish reach maturity in months and organisms can reproduce multiple times per year, a disturbed area can be recolonized rapidly by larvae settling from the surrounding intact habitat. A coral reef damaged by a storm or bleaching event can recover in years or decades. But in Antarctic waters, where organisms take years or decades to reach maturity and may reproduce only once or twice in their entire lifetimes, recovery from disturbance operates on timescales that challenge human comprehension. An area of Antarctic seafloor scraped clean by a grounding iceberg may remain essentially barren for decades before the first larvae settle and begin the slow process of community reassembly. Studies of recolonization in Antarctic waters have yielded sobering results. Experimental panels placed on the seafloor at various locations around Antarctica and left for years showed almost no settlement in the first year, sparse colonization after 3 years, and even after 2 decades of exposure remained far less densely colonized than equivalent panels in temperate waters would become in a single season. The organisms that did colonize, mostly ascidians and spirobid worms in the studies conducted so far, grew so slowly that coverage remained below 5% even after 20 years. If these are the colonization rates for relatively simple organisms like worms and sea squirts, how long would it take for a complex carnivorous sponge to establish itself, grow to maturity, and begin reproducing? The answer is probably measured in decades at minimum, possibly much longer. This extremely slow pace of life and recovery makes Antarctic ecosystems particularly vulnerable to disturbance, especially to repeated or widespread disturbance that does not allow sufficient time for recovery between events. Natural disturbances like iceberg scouring have always been part of the Antarctic marine environment, and organisms have evolved strategies to cope with them through dispersal, recolonization, and persistence in refugia that escape the worst impacts. But human activities can impose disturbances that differ in their spatial scale, temporal frequency, or type from anything these organisms have experienced in their evolutionary history, potentially overwhelming their adaptive capacity. Bottom trawling, the practice of dragging heavy nets across the seafloor to catch fish and other commercially valuable species, is perhaps the most destructive human activity impacting deep-sea ecosystems worldwide. The heavy doors and chains of a trawl net scrape across the seafloor, crushing and burying organisms, removing three-dimensional habitat structures, and homogenizing what was previously a complex patchwork of different communities and microhabitats. In areas where trawling is intensive, the seafloor can be passed over by trawls dozens or even hundreds of times per year, creating a state of chronic disturbance where communities can never develop beyond the earlier stages of succession. Around Antarctica, bottom trawling has been limited but not absent. Fishing for Antarctic toothfish, a large and valuable predatory fish that can live for 50 years and grow to over 2 meters in length, has used bottom trawls in some areas, particularly around subantarctic islands like South Georgia and Heard Island. The Convention for the Conservation of Antarctic Marine Living Resources, CCAMLR, the international body responsible for managing fishing in the Southern Ocean, has implemented restrictions on bottom trawling in many areas after recognizing the damage it causes to benthic communities. But enforcement is challenging given the vastness of the ocean and the remoteness of Antarctic waters, and illegal, unreported and unregulated fishing remains a problem. Deep-sea mining represents another potential threat, one that has not yet materialized in Antarctic waters, but looms on the horizon as technology advances and terrestrial mineral resources become scarcer. The seafloor contains valuable mineral deposits including manganese nodules, cobalt-rich crusts, and massive sulfide deposits associated with hydrothermal vents. These deposits have formed over millions of years through slow accumulation of minerals, precipitating from seawater or hydrothermal fluids, and they support unique communities of organisms found nowhere else. Mining these deposits would destroy the habitat entirely, removing the physical structure on which organisms settle and live, and the timescales for recovery would be geological, measured in millions of years, far longer than any planning horizon relevant to human affairs. The Antarctic Treaty System, which governs human activities in Antarctica and its surrounding waters, includes provisions for environmental protection that currently prohibit mineral resource extraction south of 60 degrees south latitude. But the treaty is not permanent, it comes up for review periodically, and there are nations and corporations that would like to see these protections weakened or removed to allow exploitation of Antarctica's resources. The debate over whether to permit mining in the Southern Ocean will intensify in coming decades as demand for minerals grows and as climate change makes Antarctic waters more accessible by reducing sea ice extent and duration. Climate change itself poses perhaps the greatest long-term threat to Antarctic marine ecosystems, including the carnivorous sponges that are the subject of this story. The Southern Ocean is warming, not as rapidly as some other parts of the world's oceans, but warming nonetheless. Surface temperatures in some regions have increased by more than 1 degree Celsius over the past 50 years, and this warming is penetrating to deeper waters through changes in ocean circulation. Antarctic bottom water, that dense cold water that fills the abyssal ocean basins, is becoming slightly warmer and slightly less dense as surface warming affects the formation regions where this water sinks. For organisms adapted to extremely cold temperatures, even small increases in temperature can be physiologically stressful. Antarctic fish, for instance, have antifreeze proteins in their blood that keep them from freezing at temperatures below 0 Celsius. But these same proteins make them vulnerable to even slight warming, because they have evolved to function optimally in an extraordinarily narrow temperature range. If water temperatures rise by 2 or 3 degrees, which is well within the range projected by climate models for this century, many Antarctic fish species may be unable to survive. The cascading effects through food webs could be profound, affecting not just the fish themselves but everything that eats them, or competes with them, or depends on them as predators to control populations of their own prey. Carnivorous sponges, being sessile and unable to migrate to cooler waters if their habitat warms, would be similarly trapped if temperatures rise beyond their tolerance limits. We do not know what those limits are because we have not yet conducted the experiments needed to determine them. But based on the general principle that organisms adapted to stable, extreme environments tend to have narrow thermal tolerance ranges, it is reasonable to predict that Antarctic carnivorous sponges cannot tolerate much warming before their cellular machinery begins to malfunction. Enzymes will denature, membranes will become too fluid, metabolic pathways will become unbalanced. The sponges may be able to acclimate to gradual warming through physiological adjustments, or they may be able to adapt through natural selection if warming is slow enough that evolution can keep pace. But there are limits to both acclimation and adaptation, and if those limits are exceeded, species will go extinct. Ocean acidification adds another dimension to the climate change threat. As atmospheric carbon dioxide concentrations increase, the oceans absorb some of that carbon dioxide, currently taking up about 1/4 of all the carbon dioxide emitted by human activities. When carbon dioxide dissolves in seawater, it forms carbonic acid, which increases the acidity of the water and reduces the concentration of carbonate ions. This has profound implications for organisms that build shells or skeletons from calcium carbonate, as the reduced carbonate availability makes it more difficult and energetically expensive to precipitate calcium carbonate from seawater. Carnivorous sponges build their spicules from silica rather than calcium carbonate, so they are not directly affected by the reduction in carbonate availability. However, they may be affected indirectly through changes in prey populations. Many of the small crustaceans that carnivorous sponges feed on have calcium carbonate exoskeletons and may be vulnerable to ocean acidification. If copepod and amphipod populations decline because acidification makes it harder for them to build and maintain their exoskeletons, carnivorous sponges will have less food available, potentially leading to slower growth, reduced reproduction, or increased mortality during the periods between prey captures. The compound and synergistic nature of these threats makes predicting the future of Antarctic ecosystems extraordinarily difficult. Warming, acidification, changes in sea ice extent and duration, alterations in ocean circulation patterns, increasing human presence and resource extraction pressures, and the introduction of invasive species as ships break through formerly ice-locked waters, all these factors interact in complex and often non-linear ways. An ecosystem that might be able to cope with any single stressor in isolation may collapse when multiple stressors compound each other's effects. And the extremely slow recovery times characteristic of Antarctic ecosystems mean that by the time we recognize that a threshold has been crossed and serious damage has occurred, it may be too late to prevent that damage from persisting for centuries or millennia. This is why the discovery and study of organisms like the deathball sponge is so important, not just for satisfying scientific curiosity, but for providing the baseline data and ecological understanding needed to inform conservation and management decisions. We cannot protect what we do not know exists. We cannot make informed decisions about acceptable levels of human impact if we do not understand how ecosystems function and how they respond to disturbance. And we cannot predict future changes if we do not have historical data showing what was present and in what abundance before the changes began. The biodiversity of the Southern Ocean is only now beginning to be adequately documented. Until recently, most of what we knew about Antarctic marine life came from a few well-studied locations around research stations or from occasional expeditions that collected specimens in a haphazard and opportunistic manner. The Census of Antarctic Marine Life, a major international initiative that ran from 2007 to 2011 as part of the larger Census of Marine Life, represented the first systematic effort to document what lives in the Southern Ocean, where it lives, and how much of it there is. The results of CAML, as the project is known, were simultaneously enlightening and humbling. Enlightening because they revealed an Antarctic marine fauna far richer and more diverse than most scientists had imagined, with thousands of species previously unknown to science being discovered in just a few years of intensive study. Humbling because they made clear just how little we still know, how many blanks remain on the map of Antarctic biodiversity, how many regions have never been sampled, how many groups of organisms remain poorly known or entirely unstudied. The Andeep cruises, Antarctic benthic deep-sea biodiversity, which focused specifically on deep-sea fauna in the Scotia and Weddell Seas, discovered 585 previously undescribed species of isopod crustaceans alone, potentially adding 20% to the known arthropod diversity of the region in a single series of expeditions. If isopods, which are relatively well studied compared to many other groups, can yield so many new species from limited sampling of limited geographic areas, then the true diversity of Antarctic marine life must number in the tens of thousands, or perhaps hundreds of thousands of species, most of them still unknown to science. The geographic distribution of sampling effort is highly uneven, reflecting the logistical challenges of working in Antarctic waters and the locations of research stations and regular ship routes. The Scotia Sea, Weddell Sea, and Ross Sea have been sampled more intensively than other regions because they are more accessible and have been the focus of more research programs. The Amundsen Sea, by contrast, remained biologically unexplored until 2008, when the first benthic samples were collected during the BioPearl expedition, and even now remains one of the least known parts of the Antarctic margin. Vast areas of the Antarctic continental shelf and slope have never been sampled at all, and the abyssal plains that make up most of the Southern Ocean floor remain almost entirely unknown. This patchy and biased sampling creates significant challenges for understanding large-scale biogeographic patterns and for making predictions about how climate change and other stresses will affect Antarctic ecosystems. We know that different regions of the Antarctic margin harbour different faunal assemblages, with some species being widespread while others are restricted to particular sectors or depth zones. We know that depth is a major factor structuring benthic communities, with shelf, slope and abyssal communities being quite distinct from each other. We know that the Scotia Arc, that chain of islands and submarine ridges connecting South America to the Antarctic Peninsula, plays an important role as both a dispersal corridor and a barrier to dispersal, depending on the group of organisms and the direction of movement being considered. But we do not know how much of the apparent endemism and restricted distribution of Antarctic species is real versus artifactual, a result of having not yet looked in enough places to find the full range of each species. Every new expedition discovers species previously known only from one or two locations now found in third or fourth locations, sometimes thousands of kilometers from where they were first collected. Are these species genuinely widespread but patchily distributed, or are they continuously distributed but patchily sampled? The answer matters for conservation because species with restricted ranges are generally at higher risk of extinction than species with wide ranges, but we cannot assess range sizes without adequate sampling across the full potential range. The deathball sponge provides a perfect example of these uncertainties. It was discovered during a single expedition to the South Sandwich Islands, collected from a few sites within a limited area around volcanic seamounts. Is it restricted to this region, or does it occur elsewhere in the Southern Ocean wherever appropriate habitat exists? Is it associated specifically with volcanic seamounts, or does it also occur on non-volcanic seafloor? Is it restricted to particular depth ranges or temperature regimes, or is it more broadly tolerant of environmental variation? We can begin to answer these questions through genetic analysis, comparing DNA sequences from the South Sandwich specimens to tissue samples from other carnivorous sponges collected elsewhere in the Southern Ocean, looking for matches that would indicate the species has a wider distribution than its initial discovery suggests. But definitive answers will require more expeditions, more sampling, more years of patient work accumulating the observations needed to fill in the map. The taxonomic work of describing new species and determining their relationships to previously known species is itself a massive undertaking that proceeds at a pace constrained more by the availability of expert taxonomists than by the rate of specimen collection. The camel expeditions brought back hundreds of thousands of specimens representing thousands of species, many of them new to science. But describing a new species properly requires detailed examination under microscopes, comparison with type specimens of related species held in museum collections around the world, creation of detailed illustrations and photographs documenting diagnostic characters, extraction and sequencing of DNA for molecular phylogenetic analysis, and publication of formal descriptions in peer-reviewed scientific journals. A single species description can take months or years of work by a taxonomic specialist, and there are only a few hundred such specialists worldwide for all the groups of marine invertebrates combined. As a result, there is a massive backlog of undescribed species sitting in museum collections, waiting for someone to have the time and funding to study them properly. Some expeditions from the early 1900s brought back specimens that still have not been fully described and catalogued, more than a century after they were collected. This is not because the specimens are unimportant or uninteresting, but rather because taxonomy is slow, meticulous work that requires specialized expertise, and because the number of people trained in taxonomic methods has declined in recent decades, as funding agencies and universities have shifted priorities toward molecular and experimental biology and away from descriptive natural history. The death ball sponge will eventually receive a formal scientific name, following the rules of zoological nomenclature that govern how species are named and classified. That name will consist of two parts: the genus name Chondrocladia, which it shares with dozens of other species in the same genus, and a species name that will be unique to this particular form. The species name might commemorate the location where it was discovered, or the ship and expedition that collected it, or a person who has made significant contributions to sponge biology or deep-sea exploration. Once the species is formally described and named, that name becomes part of the permanent scientific literature, a reference point that other scientists can use when they encounter similar sponges in their own work, a building block in the larger structure of biological knowledge. But beyond the practical necessity of names and classifications and formal descriptions lies a deeper question about what we are really doing when we explore and document biodiversity. At one level, we are simply making lists, catalogues of what exists, where it lives, what it looks like. This is the fundamental task of natural history, the descriptive science that underlies all other biological disciplines. You cannot study how organisms function, how they evolve, how they interact with each other and their environment, until you know what organisms exist and can reliably tell one species from another. Natural history provides the foundation on which everything else is built. But at another level, the exploration of biodiversity is an encounter with otherness, with forms of life that operate according to rules different from those governing our own existence. When we discover a carnivorous sponge, an animal that has no nervous system, no muscles, no ability to move, yet nonetheless functions as an effective predator, we are confronting the fact that intelligence and nervous systems are not prerequisites for solving the problem of obtaining food. Evolution has found a way to be a predator without any of the features we associate with predation. This challenges our assumptions about what is necessary and what is contingent, about which features of life as we know it are universal requirements, and which are merely the particular solutions that happen to evolve in our own lineage. The same is true when we encounter the extreme environmental conditions of the deep sea, and realize that organisms not only survive there but thrive, that what seems impossibly hostile to us is simply home to them, that our own comfortable environment of warm sunlit air at sea level pressure is no more fundamental or privileged than the frigid darkness of the abyssal seafloor. This perspective, this recognition that our way of being alive is just one option among countless others, is philosophically profound. It undermines anthropocentrism at a visceral level by showing us organisms whose existence is so different from ours that we struggle even to understand how they can be alive. Yet there they are, growing and reproducing and adapting and evolving, doing all the things that life does, just doing them differently. In the history of science, many of the most important advances have come from discovering that what we thought was universal was actually parochial, that what we thought was necessary was actually contingent. The Copernican revolution showed that Earth is not the centre of the universe, but rather one planet among others, orbiting an ordinary star in an ordinary galaxy. The Darwinian revolution showed that humans are not a special creation, but rather one branch on the tree of life, related by descent to all other organisms. The modern synthesis of evolution and genetics showed that the mechanisms of heredity and variation are universal, operating the same way in bacteria and humans and everything in between. Each of these advances required letting go of assumptions about human centrality and specialness, required accepting that we are part of nature rather than apart from it. The study of biodiversity, and particularly of organisms like deep-sea carnivorous sponges that seem so alien to our experience, continues this process of decentralization and humility. It shows us that life can be radically different from anything in our direct experience while still being life, still following the same basic thermodynamic and evolutionary principles that govern all biological systems. This recognition is both intellectually exciting and emotionally challenging. Exciting because it means that life as a phenomenon is more flexible, more creative, more diverse in its solutions than we could have imagined without seeing the actual organisms that evolution has produced. Challenging because it requires us to abandon cherished assumptions and familiar categories, to recognize that our intuitions about what is possible or impossible in biology are often wrong, limited by our narrow experience as one type of organism living in one type of environment. Consider what the carnivorous sponge teaches us about intelligence and behaviour. We normally think of predators as smart, as requiring sophisticated sensory systems to detect prey, neural circuits to process that sensory information and make decisions about when and how to attack, and muscular systems to execute those decisions through coordinated movement. The lion stalking the gazelle, the eagle diving toward the rabbit, the octopus extending its arms to grab a crab, all these behaviours seem to require a level of neural complexity and behavioural flexibility that is simply absent in sponges. Yet the carnivorous sponge is also a predator, captures mobile prey, digests them, obtains nutrients that allow growth and reproduction, accomplishes all the functional outcomes of predation without any of the neural machinery we associate with predatory behaviour. What this shows is that the problem of being a predator, of obtaining food by capturing and consuming other organisms, can be solved in multiple, fundamentally different ways. One solution involves brains and muscles and complex behaviour, the solution that most predators have adopted. Another solution involves passive trapping using mechanical structures that require no active control, the solution that carnivorous sponges have adopted. These solutions are not interchangeable, they work under different ecological circumstances and have different costs and benefits. But they are both valid solutions to the same fundamental problem, and the existence of both demonstrates that there is no single right way to be a predator.